Okay, I got the recording going. Okay, this lab follows very much in line with the previous lab. It's new cells, but now it focuses on meiosis. So this hopefully will help you go through some review for what we're going to cover in Chapter 9, which has to do with genetics, patterns of inheritance, okay? So you can see here a lot of information you'll need for the lab report is here in the PowerPoint, as well as you may need to use your notes or your textbook, okay, to help you fill out everything, to answer every question. It talks about here the purpose of this type of division, cell cycle including meiosis, okay, is the production of gametes that have to be haploid. Now, you got to keep in mind the parent cell that enters this type of cell division is actually a diploid cell. Okay, so this is where you're going to be for this lab. Let me get the little hand here. This is where you're going to be for this lab. You're in this area right here, okay? So when you answer your questions, you got to keep your mind on here. You got to think about, we're talking about what's happening in the of males and the ovaries of females. This is where we have a type of the cell division or cell cycle that includes meiosis occurring. Okay, we start off with parent diploid cells, okay, but they're going to divide into haploid gametes. Okay, I'm getting a message that connection is poor, so hang in there. This is being recorded. I can already tell by the fact the little hand's not keeping up with me. And you can see here these cells are haploid here. Now, meiosis itself is division of the nucleus. So this is a reminder. So imagine you're looking at a nucleus here, and we start off with a parent cell that is diploid. That means its nucleus is diploid. It contains pairs of chromosomes. Most are homologous pairs. Okay. We talk about interphase. You're going to get another question on interphase again. Should be very comparable to what you answered for this last lab with mitosis. Okay. Where every pair of homologous chromosomes or every pair of chromosomes is undergoing DNA replication. So we exit S phase with sister chromatids. But then this is where the difference comes in between mitosis and meiosis. Okay. With mitosis, you're basically talking about a somatic parent diploid cell, 2N, dividing into two identical daughter diploid cells, 2N. Okay. They have the same number of pairs of chromosomes, the same genetic information. That's why the daughter cells are identical. It's not just how they look. It's what they contain in their DNA. But these two daughter cells are also identical to the parent cell from they came from who they inherited the DNA with the genetic information from. Okay, this second type of cell division, the purpose is reproduction. Okay, and we want to make sure that when dad produces sperm, these sperm are haploid. They only carry one set of chromosomes, N equals 23. And when mom produces her eggs through this type of cell division, that they're also haploid. Okay, N equals 23. That way, if the sperm fertilizes the egg, N plus N reestablishes our diploid state in the offspring, 2N. Okay, so the organism has to be diploid, but in order to produce another generation of offspring that are also organisms are diploid, we have to produce haploid cells. So here it's showing you once every pair of chromosomes has been replicated and transformed into sister chromatids. For the first round of meiosis, okay, we have what we call segregation. Sometimes they'll say separation. In other words, what will happen during meiosis 1, well, it's not, the focus isn't just nuclear division, going from one nucleus to two nuclei, okay? But the primary focus is going from a diploid state, where we have pairs of chromosomes, to haploid states, okay? Then the purpose of meiosis 2, well, the problem with meiosis 1 is, yes, we have haploid nuclei, but they contain sister chromatids. If you recall, we went over at the end of today's lecture a process called aneuploidy, okay, where we have non-disjunction occur. Sister chromatids are either not segregated or separated from each other, resulting in haploid nuclei, or the sister chromatids themselves are not separated in the daughter chromosomes. Okay, well, if everything's going properly, going through meiosis 1, we're going from a diploid nucleus to two haploid nuclei. The problem is they have sister chromatids. We never want our final product to be a gamete, a sperm or an egg that has a sister chromatid or inherited DNA directly from or in the form of a sister chromatid. So the purpose of separate the sister chromatids into daughter chromosomes. So if you look over here to the left, we started with the parent diploid cell with the diploid nuclei. It had pairs of chromosomes. We finish with haploid gametes that have haploid nuclei, and they're only carrying one set of chromosomes. Now, Again, it's going to ask you questions having to do with S phase. Please pay attention to the questions. If I ask you for the phase, okay, don't just write in chromosome duplication and sister chromatids. Make sure you put down what phase this is occurring in also. Okay. Now it's going to be more complicated with meiosis. Now I condensed this. There are 2 rounds of nuclear division for this second type of cell division. Okay. You're going to be asked questions again, just like last week. Label the phase that is given for meiosis 1, or label the phase that is given for meiosis 2 and give a description. So you're not going to be able to copy this directly. You're going to want to feed off of your notes in lecture to help you differentiate between the two. Cytokinesis, however, does not change. It's always where the cell membrane pinches inward, dividing one cell into 2 cells. Except now, I didn't emphasize this, now in this type of cell division involving nuclear division called meiosis, the haploid gametes that are produced at the very end, they're not identical daughter cells. That's why we call them haploid gametes. So if you see something in the text that says daughter cells, don't use that term. So what I've done is I've given you the phases, such as interphase, and I put down it occurs just one time. Therefore, I only ask you about it one time. Then here I put down meiosis 1 with nuclear division, but then this first round of the meiotic phase ends with cytokinesis also. Then I give you another slide, but notice it says meiosis 2. We have a second round of nuclear division. Notice how it begins with 2 nuclei, or 2 cells versus 1, okay? But again, we're going to have cytokinesis occurring at the end where we get a total of, to put it simply, 4. Now there's a little bit of differentiation between men and women as far as the mammals go, but we'll just say we end with 4. This is just talking to you about in meiosis 1, we get a process called independent assortment, okay? The red and the blue, keep this in mind, remind yourself, the red and the blue colors, what do they represent? Maternal DNA versus paternal DNA. In other words, maternal chromosomes versus paternal chromosomes. Remember, the majority of the cells that make up your body are somatic diploid cells, 2N, okay? 1 set of 23 came from your mom. Those are your maternal chromosomes. The other set of 23 came from your dad. Those are your paternal chromosomes. This is why they give you different colors. Now, what you pass on to your children, it could be a mix of the maternal and paternal DNA that you pass on. So they're showing you this here, and this is what they're referring to as independent assortment. As usual, like before, okay, I posted a Word doc, use the Word doc or convert it to a PDF like many of you have. I've had some posted in Google's Drive, which I've been able to download it in a Word document. That's 3 different ways, okay? Double check your attachment, please. Okay, like I said, I got one attachment today, and it had a page that had nothing to do with the lab report, and I thought to myself, I hope I don't have a virus on my computer. Knock on wood, everybody. Just like before, okay, it's due on the 14th, next week, a week from today at 9:30 a.m. There are no late submissions, and I'm going to keep saying this because homework and exams are going to be coming out. Homework this week and exam, your third exam, I think, is roughly 2 weeks from today. Okay, late is late. I don't care if it's a minute late. Late is late, so make sure you stay on top. Most of y'all, at least that are here, okay, many of y'all are doing a great job of getting in your reports and getting them in that are a pretty good state. Also, as far as returning the reports, okay, I've got a lot of work to grade between 4 different classes. Okay, so I will start trying to post some grades update the grades this weekend. But as far as getting back lab reports, I may not get those back right away, but you don't have a lab practical, everybody. Now, homework, I definitely will put the effort in to get those back to you for your exam, okay? All right, so it falls in line with the same thing as a previous lab that was due today, okay? It follows the same format. Okay, are there any questions? You can use the chat board if you want, if you have any questions, unless you like the sound of your voice, you can use the mic. Okay, if there are no questions, then I'm going to ask you to leave the session. I'm going to stop the recording.